In the chapter of Cell, the unit of life, we will discuss about differentiate between plant and animal cell, sandwich model, unit membrane model. So firstly, we will talk about differentiate between plant and animal cell. So here, the first point is usually they are larger than animal cell, and animal cell usually smaller than plant. So the first differentiate point is that plant cell is large in size and animal cell is small in size. Number two, cell wall present in addition to plasma membrane and consists of middle lamella, primary and secondary wall. So as we know that in plant cell wall is present and cell wall is made up of cellulose, whereas in case of animal cells, cell wall is not present, only cell membrane is present. Cell membrane is also present in plant cell, but cell membrane is present below cell wall, and the cell wall consists of middle lamella, primary and secondary wall. Means cell wall has three layers. So this is the middle lamella. So middle lamella is present between two cells, and here primary layer is present. So this is primary layer, and here secondary layer is present. So three layers are present in cell wall. Next is chloroplast. Plant cell chloroplast is present because in chloroplast green color is present, and green color is because of chlorophyll, and that's why the plant cells are green in color because of presence of chlorophyll in chloroplast, whereas in animal cell chloroplast is absent because in case of animal cell the color is not green, that's why, because chloroplast is absent. Third, another point is vacuole is large and permanent, whereas in animal cell vacuole is small and temporary, and many vacuoles are present. So here you can see this is the vacuole. Here the one large vacuole is present and it is in the center and is permanent, and because of vacuole the plant cell is turgid because here the water, mineral and salt are present, and because of that the plant cell is turgid because of vacuole, and vacuole is large and in the center, whereas in case of animal cell vacuole are small in size and many in number, but they are temporary, they are not permanent. Next point is tonoplast present around vacuole. Means if this is a vacuole, outside membrane is present, and this membrane in case of plant cell is called tonoplast, whereas in case of animal cell membrane is not present and vacuole are temporary, that's why tonoplast is absent. Centrioles absent except motile cells of lower plants. In lower plants centrioles are present, but in higher plant, in higher plants like in angiosperm, gymnosperm, centriole are absent, and whereas in case of animal cells centrioles are present, and centrioles are responsible for formation of spindle fibers. Next is nucleus present along the periphery of the cell, because in plant cell vacuole is present in center, and because of that nucleus is pushed towards the periphery. Here nucleus is present towards the periphery, whereas in case of animal cell vacuoles are many in number and they are small and for a short period of time, and now there is no such problem, so nucleus is in the center. Lysosomes are rare, means lysosomes are very less, but in animal cell lysosomes are present. And next is storage material. In plant cell storage material is starch grain, whereas in animal cell storage material is glycogen or glycogen granules. Next is we are going to start plasma membrane, and in the plasma membrane is also called cell membrane. And composition: cell membrane is made up of or is composed of protein and phospholipid. In case of phospholipid, means lipid with lipid phosphate group is attached, like this is the lipid molecule. Here head is present, here the tail is present, and here the phosphate is present. So this is the phospholipid molecule, and this is the head portion, and head portion is hydrophilic, whereas this is the tail portion, and tail portion is hydrophobic. So hydrophilic means water loving and hydrophobic means water repellent. Hydrophilic means from this portion water molecules can pass, whereas from this portion water molecules cannot pass. So this is the structure, and here the lipid layer is present. So this is the lipid layer, and these both are lipid layers. So you can say in plasma membrane or cell membrane, lipid is bilayer, means two layers are present. And plasma membrane is selectively permeable membrane, means plasma membrane allows the entry of selective substances. For example, here this is the plasma membrane, and here the pores are present. So here some substances are present, like this molecule is present, this molecule is present, different size molecules are present. So in case of selectively permeable membrane, means plasma membrane allows the entry of selective substances which are beneficial for plants, whether they are large in size or they are small in size. For example, this is the pore size, and the pore size is very small. So in that case, this molecule can be taken in and this molecule can be taken in. But this molecule, this is large in size but is not required by the plant, so this molecule will not be taken in by plasma membrane. But this molecule is also large in size but is required by by plasma membrane, so this molecule is taken in. So how this molecule can be taken in? So in that case, there is a process of phagocytosis. So in case of phagocytosis, here the vesicle like structure is formed, like here the cup shaped structure, vesicle shaped structure is formed, and this substance or this molecule is taken in, and after that this vesicle is pinched in. So in that case, molecule with the large size is taken in. And how the molecule with the large size is taken in? So here another terminology is semi-permeable membrane. Semi-permeable membrane means the plasma membrane allows the entry of any kind of molecule, whether they are beneficial or non-beneficial to cell. So in that case, in some books there is a terminology, they have used the selectively permeable membrane terminology. In some books they use the semi-permeable membrane terminology. So in case of selectively permeable membrane, means only selective substances can be taken in, but in case of semi-permeable substances, any substances can be taken in based on the pore size. Means if the substance can pass through the pore size, whether they are beneficial or non-beneficial to cell. So in that case, so we use the selectively permeable terminology when uh, plasma or cell is the part of living system. If the cell is part of living system, then we use the terminology selectively permeable membrane. But if cell is not part of living system, then we use the terminology semi permeable membrane, because at that time cell is not part of living system, so anything can be taken in, whether they are beneficial or non-beneficial, or you can say harmful. So next is properties. So plasma membrane should have three important properties. Means selectively permeable, means plasma membrane allows the entry of selective substances, but these selective substances can be water soluble or lipid soluble. Next is variable protein to lipid ratio, means there should be variability or change in protein to lipid ratio. And third is plasma membrane should be dynamic, not static. So these three properties should be present in plasma membrane, and based on these properties, the model for the plasma membrane can be accepted. Like in the later presentation, we will discuss about uh, unit membrane model, sandwich model, and fluid mosaic model, and we will tell how whether these properties are present or these properties were present in those models or not. So here the plasma membrane models, many models had been proposed for plasma membrane, but in the lecture we will discuss about only sandwich model, unit membrane model, and fluid mosaic model. and we will tell you which model was accepted and why model was accepted. So first model is sandwich model. Sandwich model was proposed by Danieli and Davison. They were two scientists. They proposed this model. So the composition of, according to them, composition of plasma membrane was protein and phospholipid. Means protein is present. If protein is present, then the protein is globular protein. Globular protein means protein is round in shape. Like in this diagram you can see protein is round in shape. Next is phospholipid. Phospholipid molecules are amphipathic. Amphipathic means both hydrophilic and hydrophobic region are present. Like this is the lipid head, and here the tail is present, and on the head the phosphate is present. So this is the structure of phospholipid. So in the last slide I told you about head is hydrophilic and tail is hydrophobic. So head portion is hydrophilic, means water loving, and tail portion is hydrophobic, means water repellent. So if in one molecule both water loving and water repellent regions are present, then such molecule is called amphipathic molecule. So phospholipid molecule is amphipathic molecule. So in the last slide I told you about three properties should be present in plasma membrane. So in sandwich model we will see these properties were present or not. The first property, so this is the structure, and here the lipid bilayer is present. So this is the head portion, this is the tail portion, and here again the head portion and the tail portion, and these two lipid bilayers face towards each other and they are attracted towards each other through weak force, and here weak force are present between two layers. So between two layer weak force is present, and this weak force is van der Waals force. So and outside the lipid bilayer protein is present, and you can see here the globular protein is present. So this is globular protein on the both side. First property was selectively permeable, means plasma membrane should allow the entry of water soluble and lipid soluble substance. So firstly we will take lipid soluble substance. So here the lipid soluble substance is present. So here the lipid soluble substance is present because here this is the lipid molecule. So lipid soluble substance can easily pass through this area, and again whether head region, tail region, and lipid soluble molecule can pass through this area. Then the lipid soluble substance has entered the intracellular cytosol. So there is no problem with the lipid soluble substance. Now we come to the water soluble substance. Here the water soluble substance, let me take another color. So here the water soluble substance. So this is the head portion, means it is hydrophilic portion, water loving, where it is the hydrophobic portion. So here the hydrophobic portion. So water soluble substance can pass through this area. Now this area, when the water molecule has come to the hydrophobic region, so here the water molecule will not pass through or will not cross this area because of hydrophobicity. So here in the sandwich model, Danieli and Davison did not explain about the pathway for water soluble substances. Next is, so first property was not explained properly, and next is variable protein to lipid ratio. And in the sandwich model, Danieli and Davison did not explain about the variability in the protein and lipid ratio. And if they did not explain the variability or variation or variable protein to lipid ratio, it means they did not explain whether this model is, or plasma membrane is, dynamic or statics. So because of that, sandwich model was not accepted, and it is not acceptable model for plasma membrane. Next is unit membrane model. So after sandwich model, now we are going going to discuss about unit membrane model. Unit membrane model was proposed by Robertson. Composition was similar, like plasma membrane is made up of protein and phospholipid. In case of unit membrane, phospholipid is empathic molecule like we discussed in the last presentation slide, but the protein is fibrous, and globular protein is rounded structure, but the fibrous protein is like this, or you can draw fibrous protein like this. So in case of there is a difference between unit membrane and the sandwich model. Firstly, the protein structure. In the protein structure, the protein in sandwich model was globular protein, but in unit membrane model the protein was fibrous protein. Now there are three properties. Now we will try to understand whether this model was accepted or not accepted based on these three properties of plasma membrane. First property was selectively permeable. Now we will start from the lipid soluble substance, because this is the lipid soluble substance. So this lipid soluble substance can easily pass through the membrane, or pass through the lipid bilayer and then the protein, and now the lipid molecule has entered the intercellular cytosol, because the lipid bilayer is composed of phospholipid, and the lipid is present, so there was no problem for lipid soluble substances. But now if we come to the water soluble substance, so here there will be the problem for water soluble substance, because here the water molecule can pass through the head region, but now the water molecule has reached the uh, tail region, and you know tail is hydrophobic in nature, and this tail will not allow or will not let the pass water soluble substances. So Robertson did not explain the path for water soluble substance, so first property was not fulfilled. Second was variable protein to lipid ratio. Like sandwich model, Robertson did not explain the variability in the protein to lipid ratio, and they also did not explain the dynamic, whether the plasma membrane is dynamic or static in nature. So that's why this model was also rejected or was not accepted for the plasma membrane.